Understanding the typical applications of a micro-high-speed centrifuge can help determine whether it meets specific experimental needs. These versatile machines are used in a variety of scientific and medical settings. Typical applications: cell separation. Description: used to separate cells from fluids or to pellet cells for further analysis. Importance: essential for preparing samples for cell culture, PCR, and other molecular biology techniques. Plasma, serum isolation. Description: centrifugation separates plasma or serum from blood cells. Importance: crucial for clinical diagnostics and research involving biochemical assays. Protein purification. Description: helps in purifying proteins by removing contaminants. Importance: important for protein biochemistry and structural studies. Nucleic acid extraction. Description: used in the purification of DNA and RNA from cells or tissues. Importance: necessary for genetic testing, sequencing, and molecular cloning. Microbial pelleting. Description: enables the concentration of bacteria or viruses from large volumes. Importance: useful for microbiological research and vaccine production. Microfluidics and nanotechnology. Description: used in the preparation of nanomaterials and microscale devices. Importance: supports cutting-edge research and development in biotechnology and materials science. The typical applications of a micro-high-speed centrifuge span across various fields, including molecular biology, clinical diagnostics, and biotechnology. Understanding these applications can help in selecting the right centrifuge. To learn more about our range of micro-high-speed centrifuges, or if you have any questions regarding their operation, please contact me through my email provided on my homepage. I will respond within 24 hours.